The functional development of the nervous system is a highly coordinated and progressive process that transforms a simple sheet of ectodermal cells into a complex, highly organized structure capable of sensation, movement, emotion, memory, and consciousness. It spans the embryonic, fetal, neonatal, and postnatal stages, involving the maturation of both structural and physiological components necessary for full neurological function. Functional development begins shortly after the formation of the neural tube. During the early embryonic period, neurogenesis occurs. Neural stem cells in the ventricular zone of the neural tube proliferate and begin differentiating into neurons and glial cells. These neurons are not yet functionally mature. For function to emerge, neurons must first migrate to their appropriate locations, differentiate into specific types, extend axons, and form synaptic connections. One of the earliest indicators of functional maturation is the emergence of spontaneous electrical activity. Even before synaptic connections are fully established, immature neurons generate spontaneous calcium transients and membrane potentials. This intrinsic activity plays a role in shaping synaptic connectivity and guiding further development. As the brain vesicles form and regionalization occurs, specific areas of the brain begin to specialize for different functions. The forebrain develops centers for cognition, emotion, and voluntary movement. The midbrain handles reflexes and visual/auditory processing. The hindbrain regulates vital functions such as breathing, heart rate, and coordination. Cranial and spinal nerves begin to form, and motor and sensory roots emerge. Motor neurons in the spinal cord begin to innervate developing muscles, allowing primitive reflex movements. By the end of the first trimester, basic flexion and extension movements of the limbs can be observed in the embryo. The peripheral sensory system also begins to mature. Sensory neurons from dorsal root ganglia send projections into the dorsal spinal cord and out toward developing skin and viscera. This allows for the formation of primitive sensory pathways, and by the second trimester, fetuses can respond to tactile stimuli. Myelination is a key part of functional development. Although oligodendrocytes and Schwann cells appear during embryonic development, active myelination of axons begins in the late fetal period and continues postnatally. Myelin increases conduction velocity and is essential for coordinated and rapid signaling. In the central nervous system, myelination begins in the spinal cord and brainstem before progressing to the cerebral hemispheres. The corticospinal tract myelinates from inferior to superior, and posterior to anterior, with the prefrontal cortex being one of the last areas to fully myelinate. Synaptogenesis, the formation of synaptic connections between neurons, starts in utero and accelerates during the third trimester and early postnatal life. It is initially excessive, leading to a surplus of synapses. Synaptic pruning follows, eliminating weaker connections while strengthening others based on activity and experience. This process is guided by neurotrophic factors such as nerve growth factor, NGF, and brain-derived neurotrophic factor, BDNF. Functional circuits begin to emerge. Reflex arcs are among the first to become functional. The withdrawal reflex, grasp reflex, and rooting reflex are early signs of functional spinal and brainstem circuits. More complex behaviors, like coordinated eye movements, appear later as supraspinal centers develop and connect with lower centers. In the fetal brain, sleep-wake cycles begin to develop around the third trimester, and fetal EEGs begin to show distinguishable patterns. Sensory systems mature in a specific order. Touch is the earliest to develop, followed by the vestibular system, then smell and taste, and lastly vision and hearing. Postnatal development continues with rapid expansion of dendritic arbors, increases in synaptic density, and further myelination. The first year of life is characterized by rapid growth in brain size, refinement of motor skills, and the emergence of voluntary movement. Language circuits become active in the first few months, laying the foundation for speech. Executive functions and abstract reasoning develop much later, continuing through adolescence as the prefrontal cortex matures. Critical periods exist in the development of many sensory and cognitive systems. These are time windows during which experience and environmental input are essential for normal maturation. For example, visual deprivation during early life can lead to permanent deficits due to disrupted development of the visual cortex. Similarly, language acquisition has an optimal window in early childhood. Neuroplasticity is a hallmark of the developing nervous system. Immature brains are highly adaptable, capable of reorganizing in response to injury or environmental change. However, this plasticity declines with age as circuits become more specialized and less malleable. In summary, functional development of the nervous system involves a sequence of tightly regulated events: neurogenesis, migration, differentiation, axon growth, synaptogenesis, myelination, and circuit refinement. It is a dynamic process influenced by both genetic programming and environmental input. Disruptions at any stage can result in neurodevelopmental disorders, highlighting the importance of early diagnosis, intervention, and support in pediatric neurology.